everyone. I welcome everyone who has just joined us. We are very proud and happy to welcome today Dr. Maria Zimmermann Kogadeeva. I hope that I pronounced it okay. She's joining us from the European Molecular Biology Lab in Heidelberg. And she is doing research that other people are just dreaming about, which is meta everything. You know, she has a specialist degree in mathematics and systems programming that she obtained in Moscow, Russia, from 2006 to 2011. And she stayed true to herself by then going to the best systems biology labs in the world, first to Uwe Sauer and Nicola Zamboni at Zurich in Switzerland, where she did a PhD until 2016, and then did a postdoctoral fellow at Andrew Goodman's lab at Yale University School of Medicine, where she did look at high throughput screening, kinetic modeling to investigate the contribution of the gut microbiome to the metabolism of medical drugs. And then since 2019, she is at Professor Pia Borg's laboratory at EMBL, doing multi-omics integration to disentangle diet-host microbiota interactions. So together with, you know, metagenomics, drugs, diets, it's hard to imagine a field that is more entangled and more interesting than the field that Maria Zimmermann Kogadeeva is working on. So we are very pleased to have her with us today and tell us about her recent research. Thank you so much.
Thank you very much for this kind introduction. I'll now share my screen and ask you to confirm that you see it and that you also see my pointer.
Yes, we see your screen.
Thanks a lot. Okay, and again now.
Oh yeah, yep. Okay.
Okay. So thank you very much for inviting me to present my work in this Metabolomics seminar. It's the ongoing work that I've been doing here at EMBL Heidelberg, but many projects that I would like to share with you today. I started at Andy Goodman's lab at Yale University. I hope that today I can share with you how we integrate metabolomics with other omics to explore host microbiome interactions. So first of all, we come to this world practically sterile. And throughout our life, we encounter microorganisms that live in and on our bodies, from the people we interact with, environment we live in, and the food we consume. A healthy human body harbors several hundreds of microbial species in different organs, with the majority of the species residing in the gut and comprising the so-called human gut microbiome. Human gut microbiome collectively encodes 100 times more genes than the human genome. And recent advances in sequencing technologies allow us to correlate or associate different species abundances in the human gut to different aspects of human health, such as digestion, development of immune system, mental health, as well as a variety of metabolic diseases, metabolism of medical drugs, and processes associated with aging. In my research work, I mainly focus on the metabolic interactions between microbiome and the host, or interactions through metabolites or small molecules that can be consumed and produced and exchanged between the microbiome and the host. And throughout this talk, I will walk you through our attempts to investigate these three mechanistic questions of host microbiome interactions. The first question is what determines bacterial survival in the gut? The second one is whether bacteria contribute to the host metabolism, and if yes, how much they can contribute to these metabolic processes in the host. To answer the first question, we started with a genetic approach focusing on the gut microbiome, on the microbes. And this genetic approach is called Transposon Insertion Mutagenesis. And the idea is that we can generate, in a given microbe of interest, we can generate a library of genetic mutants by inserting transposons randomly in the bacterial genome, and then expose these libraries to different conditions, for example, in vitro cultivation or mouse colonization, and then use high-throughput sequencing to compare the contents of the input library and the output library and identify genes that confer fitness to these microbes. In this scatter plot here, I'm showing you the results of such analysis, where each dot here is a gene, and on the x-axis is the number of mutants in the library that was growing in vitro in laboratory conditions, and on the y-axis is the mutants grown in the mouse. And as you can see in this gray diagonal here are genes that are equally important in the gut and in vitro conditions, but there are also differences. For example, this blue gene here has many more insertions in vitro compared to the mouse gut, suggesting that in the mouse gut, the deletion of this gene is detrimental for the mutant, or as red gene here that doesn't carry any insertions neither in vitro nor in vivo, suggesting that it's an essential gene without which bacteria cannot survive at all. And I'm showing you those two genes for a particular reason. They caught our attention because they're actually homologous, which means that they are annotated. They have similar sequence, and they both annotated as Ppgpp synthase and hydrolase. So you're probably wondering, what is Ppgpp? Ppgpp is a small molecule. It's a metabolite. It is a quite well-studied bacterial alarmone. So many bacteria, including E. coli, a widely used laboratory strain, and many pathogenic bacteria make this Ppgpp molecule as a response to different types of stresses or infection. And what we realized by bioinformatics analysis is that whereas this blue gene here, BT0700 is the gene ID of this gene, it retained only part of the function of the Ppgpp synthase hydrolase, namely only synthase, so it can only make Ppgpp. But the red gene, retained both synthase and hydrolase function, meaning that it can both make and degrade Ppgpp. That explains why we can delete the blue gene, because if blue gene is deleted, the red one will take over the function. But we cannot delete the red gene because in the absence of hydrolase, Ppgpp is synthesized to toxic levels and bacteria die of Ppgpp overproduction. However, we found that if you delete both of these genes simultaneously, this mutant bacteria will survive, allowing us to investigate the role of this bacterial molecule, Ppgpp stress alarmone, in the gut. So now we have two bacterial strains, the wild type strain and a Ppgpp0 strain that is not able to produce this stress molecule, and we can test how they behave in different conditions. So in this plot, I'm showing you their survival curve of these two mutants in carbon starvation. So when we grow them in laboratory conditions, and then we remove the carbon source, and then over the course of several days, we test how many bacteria are still surviving. And as you can see, this red curve here, this is the mutant that is dying much faster than the wild type. And when we can take these two mutants and inoculate them into the mouse gut and compare also after several days how much of each mutant is retained in the mouse gut. And as you can see in this middle plot here, also when both wild type and mutant were colonizing mouse gut, their mutant was losing in competition and basically getting eliminated from the system. However, if we took either of this mutant and inoculated them into a germ-free mouse gut, so a mouse that was completely sterile and doesn't have any microbiota, we found that in isolation, each of the strain was able to colonize and grow in the mouse intestines. So to investigate what are the differences that makes Ppgpp0 strain susceptible to these different stresses, we first performed transcriptomic analysis and measured all the gene expression of this mutant and wild type strain, either after 1 hour of carbon starvation, so in the beginning of the starvation response, where we have a lot of bacteria here, or in the molecular colonization of mice, where both bacteria grow happily. And to investigate the and you can see on these volcano plots that there are quite a lot of genes that are differentially expressed between Ppgpp0 strain and wild type strain, either in the carbon starvation or in the mouse. And to investigate the processes associated with these genes, we took the metabolic network approach, where we take the metabolic network that represents bacterial metabolism or bacterial ability to convert substrates to products and produce different building blocks for its to sustain its life, and investigate the metabolic paths in this network. So here, each node is a metabolite, and each edge is a gene that is abbreviated here with their ID, and then we can project the differences in gene expression between the Ppgpp0 mutant strain and the wild type strain on this network. And as you can see here, the orange genes are the ones that were upregulated in the mutant strain, and the green genes are the ones that are downregulated in the mutant strain when colonizing the mouse. Then we can extract the metabolic subnetworks from this network that are concordantly regulated. Here we found 5 concordantly regulated metabolic subnetworks. And although here they still look like a hairball to you, when we rearrange them, according to the textbook, we find actually that there are very well-known textbook metabolic processes that distinguish Ppgpp0 strain from wild type in the mouse gut, namely the downregulation of sugar metabolism, central metabolism, and Tca cycle, and the upregulation of amino acid metabolism. When we when we mapped the genetic gene expression differences between mutant and wild type strain in the starvation condition, we found that both in the mouse gut and in vitro starvation, the differences between the mutant and the wild type strain was virtually the same. The core differences in Ppgpp0 response were the same. So this analysis, these results told us that Ppgpp induces some global transcriptional response that is similar between these very different environmental conditions, and it allows us to further investigate the much more experimentally tractable in survival in vitro condition to understand a bit more what is happening in the Ppgpp0 strain. So to do so, we're now focusing on the carbon starvation. And here, in addition to transcriptomics, to gene expression, we also measured metabolomics. And I promise you, since this slide, metabolomics will be the main tool of our analysis of bacterial fitness in the gut. So what we found, what we're looking at here is the differences in wild type strain in 1 hour starvation versus growth. So how does wild type strain regulate its metabolism in transcriptional level and on metabolite level when it starts to starve? And we compare this response to the Ppgpp0 response in carbon starvation. And although these 2 maps look quite similar, with, for example, downregulation of sugar metabolism and upregulation of amino acid metabolism upon starvation, there are very drastic differences in a part of central metabolism, which is called Tca cycle. And as you can see, the wild type strain is upregulating the several enzymes that constitute the oxidative branch of the Tca cycle. And in the wild type strain, there is an increase in the intermediates of Tca cycle succinate, and especially alpha-ketoglutarate, whereas the Ppgpp mutant strain failed to perform this regulation. So here we asked whether this increase in alpha-ketoglutarate metabolite that's produced in the wild type, whether it affects survival and how it is made under stress conditions. So to follow how alpha-ketoglutarate is made in the wild type, we used experimental technique, which is called stable isotope labeling, where we shifted bacteria growing in normal minimal media, including glucose, to minimal media including glucose that is stable carbon labeled in 6 positions. So we can then measure this intracellular metabolites over time and see how their labeled glucose is replenishing the unlabeled glucose, as shown here in the top plot. And in the downstream metabolites, we see that the glucose molecule is that this has 6 carbons is split into 2 phosphoenol pyruvate molecules that also over the course of several minutes and hours gets fully replenished by the carbon label. But what we're actually interested in is the labeling patterns in the Tca cycle, in the lower metabolism. And what we found that alpha-ketoglutarate and glutamate, what we're interested in here, have labeling patterns that are similar to the ones of succinate, which is a part of the reductive branch of the Tca cycle, and not citrate, which is a part of the oxidative cycle, that allowed us to hypothesize or provide our evidence that alpha-ketoglutarate is made in the reductive part of the Tca cycle. We also found that even in starvation, the labeling patterns of glutamate and alpha-ketoglutarate were resembling those of malate and succinate and not the ones of citrate, suggesting that alpha-ketoglutarate is made by wild type and by Ppgpp0 strain from the reductive part of Tca cycle, both during the growth and in carbon starvation. And finally, we performed our main validation experiment where we added alpha-ketoglutarate to the starving cells and monitored whether addition of alpha-ketoglutarate would help them to survive longer. And as you can see here, the wild type strain was surviving with and without alpha-ketoglutarate equally well, but the mutant strain that was drastically different and dying much faster than the wild type was partially rescued by addition of alpha-ketoglutarate, suggesting that alpha-ketoglutarate alone confers quite a lot of metabolic regulation in carbon starvation in the gut bacteria. So to summarize this part, I showed you that Ppgpp metabolite is required for colonization of the gut of freely fed mice and also confers fitness in carbon starvation in the gut bacteria. And by comparing transcriptional responses between in vitro and in vivo conditions, we could identify co-response that was actually similar, suggesting that Ppgpp invokes the same response regardless of condition. And finally, at least for carbon starvation, we identified alpha-ketoglutarate as a co-regulatory metabolite, helping bacteria to survive longer the carbon starvation. And this alpha-ketoglutarate response is dependent on the Ppgpp0 activation. And now I would like to shift gears from focusing on bacteria and bacterial fitness in the gut to answering the question of whether bacteria in the gut contribute to the host metabolism. And we started answering this question with the focusing on medical drugs. Medical drugs are particularly convenient experimentally because they're produced neither by the host nor by the microbes, but they can be introduced into the system into a more controlled way. So we first started with one single drug, brividine, which is an antiviral compound, and it's known to be converted both by the host and by the microbiome into a potentially conditionally toxic metabolite bromovinyl uracil, or BVU. And we confirmed that both the host and the microbe can perform this conversion by incubating the drug with human and mouse liver cells or human and mouse gut contents, and showing that over time, both host and the microbes, both mouse and human, and both mouse and human microbiome can convert the drug to the toxic metabolite. Then we asked whether this microbial ability to metabolize brividine also plays a role in vitro. And here again, we use this notobiotic mouse model, which is a absolutely sterile mice that don't have any microbes neither on them nor in them, and they live in the bubble. And we can introduce any microbiome we want in these microbes. So here we compare the brividine patterns of brividine metabolism in germ-free mice that are completely sterile and in colonized mice in blue that have a normal microbiota. We measured drug and metabolite concentrations over time until 9 hours after drug administration. And we found that in the colon, in the large intestine of germ-free mice, we find much higher levels of the drug, whereas the drug levels in the serum is quite comparable between germ-free and colonized mice. On the contrary, for the toxic metabolite, we found that the colonized mice had much higher levels of this metabolite in the blood, whereas the levels in the large intestine were fairly comparable between germ-free and colonized mice. So here we were wondering, what are the processes that make these differences in drug and metabolite patterns in vivo, and whether we can quantify host and bacterial contributions to brivudine to BVU conversion in the organism. To do so, we developed a physiology-based pharmacokinetic model describing different processes of drug metabolism in the body. So when drug enters the body, it can be either absorbed in the small intestine into the systemic circulation or serum, where in turn it can be converted by the host into metabolite, or it can be propagated down the gastrointestinal tract and getting eliminated in the system. We describe these processes with a series of ordinary differential equations that, for example, in this case, describe that the concentration of drug in serum is proportional to the drug absorption from the small intestine, the host metabolism of drug, and the elimination of drug from the system. In colonized mice, however, the drug that reaches large intestine encounters bacteria that live in the large intestine, and this bacteria can in turn convert drug to metabolite, and this metabolite can either be propagated down the gastrointestinal tract or absorbed in the systemic circulation. And we used the data from germ-free mice to estimate the parameters of host drug metabolism, and the data from large intestine alone of the colonized mice to estimate the parameters of microbial drug metabolism. And then we use this model to predict how much of the toxic metabolite will be produced by the host and how much will be produced by the microbe in the colonized mice. And when we overlaid this total predicted exposure to the toxic drug metabolite, we found that the predicted exposure corresponded very well to the measured experimental concentration of the toxic metabolite in the serum. And this model allowed us to quantify that bacteria contributed 70% of the toxic metabolite exposure in the total organism. Having this model at hand, we can now investigate how different parameters of this system affect exposure to toxic metabolite. For example, here we can vary the ratio between host drug metabolism and bacterial drug metabolism and model what will be their metabolite profile in the systemic circulation and how much of it will be contributed by bacteria here in the red dashed line. We can also investigate this system when we vary several parameters at the same time. For example, the surface plot here shows how at different values of host drug metabolism or bacterial drug metabolism, how much of the toxic metabolite will be produced in the systemic circulation on this z-axis, and how much of it will come from bacteria, which is indicated here by the color. So, for example, when bacterial coefficient is close to 0, then all their exposure of host to toxic metabolite will be coming from the host, and vice versa. We also showed this model can be generalized to other drugs, and showed for 2 other drugs, soribudine, that has similar chemistry as brivudine, and clonazepam, that is a very different drugs, that even also in this case, our model is applicable, and our model can help us to quantify that bacteria in the gut contribute the majority of toxic metabolite exposure in the systemic circulation. So inspired by these findings, we were wondering how often actually bacteria can contribute to drug metabolism. So we set out to systematically assess the potential of gut bacteria to metabolize medical drugs. To do so, we performed a large-scale screen where we incubated 76 bacteria isolated from the human gut against 271 human-targeted non-antibiotic clinical drugs and measured drug concentration after 12 hours of incubation with liquid chromatography coupled mass spectrometry. We found that 2/3 of the drugs we studied were metabolized by at least one bacterial strain from our system. As shown in this heat map, the lighter the color, the more drug was metabolized after 12 hours of incubation. And we found that the patterns of bacterial metabolism shown here in this heat map actually separate bacteria based on their phylogenetic relationship, confirming that bacteria that are similar in their genetic potential and their genomes have similar capacities to metabolize medical drugs. On the other hand, we also found that drugs that clustered together and were metabolized likely in a similar way also shared chemical structures, chemical substructures, as indicated here by this red acetyl groups highlighted for the 5 drugs metabolized by Bacteroides species. So here we set out to identify products of microbial drug metabolism. To do so, we went back to our collected data set and took advantage of our large-scale screen by comparing which metabolites are present in the pools that contain drugs versus all other samples that don't contain a specific drug. And here on this volcano plot, I'm showing you the fold changes of the compound abundance against the p-value of how significant these differences are for one specific drug, diltiazem, in one specific bacteria, Bacteroides Tca Omicron. And as you can see here, there were only 2 metabolites that were significantly more abundant when diltiazem was present. One of this compound was diltiazem itself, and the other compound is a metabolite that is exactly 42.0.11 Dalton lighter than the diltiazem drug. And for those of you who are fond of chemistry, you can say, well, this looks like a loss of acetyl group, and indeed, this red acetyl group in diltiazem, we hypothesize that this is the conversion that bacteria are making and to metabolize this drug. And when we performed this analysis for all drugs that were metabolized by our bacteria, we found that there are discrete patterns of the mass shifts between the drugs and the metabolites that we identified in this untargeted screen. So for example, here we found 9 drugs that had a metabolite that was 42.0.11 Dalton lighter than the parent drug. And here I'm showing you the structures of this drug, and you can appreciate that they all indeed have this acetyl group that can be cleaved off by bacteria. So here it suggests that bacteria can perform similar chemical reaction on these different drugs. So our next question was whether we can actually identify bacterial genes that are responsible for these chemical reactions. And to do so, we went back again to the genetic approach of genetic screens. And in this case, we used a gain-of-function library. In this experimental approach, donor DNA from bacteria of interest, for example, Bacteroides Tca Omicron, a common human gut commensal, is sheared into small pieces and expressed in a different bacteria in a heterologous host, Escherichia coli, that is actually not able to metabolize drugs. So then we have a large library of E. coli mutants that all contain a random part of the donor DNA, and we can then incubate this library with the drugs that donor bacteria is metabolizing and test whether E. coli gain the capacity to metabolize these drugs. And here I just show you one example for an illustration. So this heat map represents 384 deep well plates. Each of this well contains an E. coli mutant with a random insertion of donor DNA. And as you can appreciate, in this 4 plates that collectively have more than 1,000 mutants, there is only one well that in which their drug concentration was decreased and the drug metabolite concentration was decreased. And this well is in the position J7 of this gain-of-function library. In this manner, we screened several dozens of thousands of E. coli strains, and we identified 4 independent strains that gained the capacity to metabolize diltiazem. And when we mapped, we then sequenced the insertion of the donor DNA in these strains, and we found that all these 4 independent strains got a insert from the donor mapping to the same genetic location in beta-iota Omicron genome, covering the gene BT4096. We then confirmed in the lab that this gene BT4096 is sufficient to metabolize diltiazem by expressing its clean copy in E. coli and showing that E. coli with this gene here in red compared to wild type gained the capacity to convert diltiazem to diacetyl diltiazem. And on the other hand, 4096 is necessary to metabolize diltiazem because when we took the donor strain and knocked out the 4096, we found that the mutant here shown in black is no longer able to metabolize drug to the desacylated form as fast as the wild type mutant can. And finally, we used germ-free mice, again, notobiotic mice, and we inoculated this mice with either the mutant or the wild type bacterium and showed that the single bacterial gene missing in the wild type bacteria drastically affects the levels of drug and metabolite in the mouse intestine. As you can see now in the mutant bacteria that's lacking this gene, there are much higher levels of the drug and much lower levels of the drug metabolite in the mouse. Diltiazem is just one example. In the same approach, we identified several dozens of bacterial genes metabolizing several dozens of drugs in beta-iota Omicron, which allowed us to build this network of drug gene metabolite connections that were all validated in vitro. And with the same approach, we also analyzed 2 other bacterial species that are common in the human gut, a Bacteroides dorii, that is a species similar to Bacteroides satiota Omicron, Ecolincella aureofaciens, which is a very different representative of the human gut. So next, we wanted to see whether the identified bacterial genes can actually explain the results that we observed in our initial screen of drug metabolism. With to test this, we went back to the results of our screen and we sorted bacteria by their ability to metabolize the drug, in this case diltiazem. The higher the bar, the more drug bacteria metabolized. And this heat map below shows the presence of the homolog to this identified 4096 gene in the genomes of this different gut bacteria. And as you can see here, only bacteria that had a homolog to 4096 were able to metabolize this drug, making 4096 a perfect predictor of whether bacteria will metabolize diltiazem or not in our 76 species screen. And here I'm showing you this statement illustrated with a ROC curve that shows that the homology to 4096 can distinguish all the metabolizers from all the non-metabolizer whole diltiazem. We found also other examples. For example, for a drug norethindrone acetate, we found 2 independent genes, one in Bacteroides satiota Omicron and one in Bacteroides dorii. These genes turned out to be homologs, and also their homology to this gene present in the genomes of other bacteria were predictive whether this other gut bacteria will metabolize norethindrone acetate or not. There are also more complicated cases. For example, for nictinidazole, we identified genes from all 3 species that were in isolation able to metabolize tinidazole. And as you can see from this heat map here, some of these genes are homologous to each other and some of them are not. And whereas each single of these genes was a pretty poor predictor of whether bacteria will metabolize tinidazole or not, the combination of these genes or the rule of whether there is a homolog to at least one of these genes in bacterial genome was a very good predictor of whether single species will metabolize tinidazole or not in our single species screen. So you're wondering, probably at this point, okay, this is all but only based on the single species level, but our real gut community contains hundreds of bacterial species. Will these findings also be relevant there? And To answer this question, we performed one more experiment in this study where we took a healthy human gut communities. So it's basically stool samples of 30 healthy humans, and we incubated this entire stool contents with diltiazem and measured diltiazem concentration over time. And we found that these 30 humans varied quite drastically in their capacity to metabolize, to convert diltiazem to deacetyl-diltiazem. So they had different rates of drug conversion. And we then performed metagenomic sequencing of these human stool samples to identify and measured the abundance of different species and also the abundance of the genes that we identified in vitro step. And as you can see from the scatter plots, their abundance of the 1496 homologs in the community is correlating with the rate of drug to metabolite conversion of the entire community, and compared to other metagenomic features such as abundance of bacterial phyla, genera, or species, the abundance of a homolog to the identified gene was correlating at the highest level with the community capacity to metabolize the drug. So in this part, I showed you how our computational approaches and genetic approaches in the gut human microbes and notobiotic mouse models allow us to quantify host and bacterial contribution to a shared metabolic process in vivo, how with the help of high throughput metabolomics, we can identify novel metabolic functions in human gut microbes, and how combining this with metagenomics and with genetic approaches allow us to identify genes in the microbes that are predictive of the community metabolic behavior of the complex human gut communities. And in the last few minutes, I want to shift gears a little bit and tell you about an ongoing project and published work that I started in Annie Goodman's lab at Yale and now continue at EMBL Heidelberg on investigating microbial contribution to the host metabolism in the context of diet. So now we are moving from a very controlled system where we're introducing the drugs and measuring their metabolism to an entire plentiful of nutrients that contained in the host diet. But to disentangle diet, host, and microbiome contributions to the host metabolites, we designed a controlled experiment in notobiotic mice, where we took germ-free mice that were completely sterile and colonized them with a defined community of common human gut microbes. So we combined this 18 common representatives of human gut in vitro, inoculated them into the germ-free mice, and then we exposed this germ-free and colonized mice to two different types of diets, a high carbohydrate diet or controlled diet here in blue, and high fat diet here in orange. And after 4 weeks on these two controlled diets, we collected metabolomic samples along different mouse tissues, as well as metagenomics and metatranscriptomics samples from the large intestine of colonized mice, where the majority of bacteria reside. With untargeted liquid chromatography coupled mass spectrometry, we could identify more than 20,000 ions along the gastrointestinal tract and other mouse tissues, and we could annotate more than 3,500 of these compounds with KEGG and human metabolite databases. And as you see on this KEGG heat map representing microbial and host metabolism, we could have a pretty good coverage of metabolites in the metabolic network. Our first question was, what is the distribution of different metabolites across the host tissues? To answer this question, we normalized abundance of metabolites along the tissues of gastrointestinal tract. We collected metabolite samples from 3 sections of the small intestine and 3 sections of the large intestine. And then we performed unsupervised k-means clustering of these metabolite patterns in the gastrointestinal tract. And we found 6 clusters that emerged and that are shown here in the top panel. And as you can appreciate, we could we could connect each of this cluster to the peak intensity of metabolite in one of the 6 fractions along the gastrointestinal tract. And here are just some examples. For example, a tyrosine, which is likely contained in the diet, had the highest abundance in the duodenum, the first part of the small intestine. Tyrolithocolate, which is a cholic acid likely produced by the host, had the highest abundance in ileum, or in the later part of the small intestine. And there were a number of metabolites that were abundant in the large intestine. And we performed this K-means clustering separately for the 4 groups of mice, colonized and germ-free on high carbohydrate and high fat diet. And we found that the same 6 clusters emerged in all 4 conditions. So then we asked whether there are specific chemical groups that are enriched in one or the other cluster. This heat map shows you the chemical group enrichment analysis. And as you can appreciate from this map, their metabolites belonging to the same group were belonging to the same cluster, regardless of colonization or diet state of the mouse. For example, metabolites that belong to the first cluster were amino acids, purines, monoacylglycerols that are likely components of the diet, so they have the highest intensity in the upper part of the small intestine. Metabolites such as bile acids or steroid glucosides were abundant in the ileum, or the later part of the small intestine, and those compounds are known to be produced by the host and released through the bile duct into small intestine to aid digestion. There were also different metabolite groups such as glycosphingolipids, bilirubins, or cholesterols that were present along the large intestine, where the majority of microbes reside. So whereas their chemical groups of metabolites had the same distribution patterns across tissues, we were wondering whether there are still differences in metabolite levels between conditions. We did found these differences, and here you can see with these 3 examples that although their spatial distribution of metabolites is the same in these 3 examples for all 4 conditions, we still find differences. For example, this metabolite 5-aminolevullinate is more abundant in the gut of germ-free mice, here in pale colors, or this metabolite porphobilinogen is more abundant in the gut of colonized mice, here in more solid colors. To quantify different factors, such as host and microbiome contribution to this metabolite profiles in the tissues, we developed a simple metabolic flux model of the gastrointestinal tract that assumes a steady state of metabolite profiles and takes into account 4 sources of metabolite abundance. The first source is intestinal flow, or just the flow of fecal matter throughout the intestine. The second source is host metabolism in the small intestine, then host metabolism in the large intestine, and microbiome metabolized in the large intestine. By fitting the parameters of the simple model to all 4 mouse groups simultaneously, we can then assess for each metabolite what are the factors mainly contributing to its metabolite profile. And here I would like to walk you through some examples. For example, leucine here has the major contributing factor to its shape in the gastrointestinal tract is host absorption in the small intestine, suggesting that this is a dietary metabolite. For 5-aminolevullinate, although the host absorption in the small intestine is still the major contributing factor of its shape, we still find a substantial contribution of host metabolism and potential bacterial consumption that would explain these differences between germ-free and colonized mice in the large intestine. And finally, for porphobilinogen, our model predicts that both host and microbes are involved in porphobilinogen synthesis that can explain these differences between the 4 mouse groups. We performed this type of analysis systematically for more than 2,500 of metabolites detected in the large intestine, but that allowed us to classify metabolites based on their main source in the large intestine. We found that almost 40% of metabolites in the along the gastrointestinal tract are mainly coming from the diet because their main contributing factor is host absorption in the small intestine. About 1/3 of metabolites are produced by the host, either in large intestine or in small intestine, and about 15% of metabolites detected in the gastrointestinal tract are potentially directly produced by the microbes. So here we ask whether we can explain the microbiome contribution to the metabolite profiles. And first, we use this modeling coefficients to define potential microbial substrates or metabolites for which microbial coefficient of metabolism predicted by the model is negative, and potential microbial products where the metabolic coefficient of bacteria is positive. And here the potential substrate, 5-aminolivulinate, that is abundant in the gut of germ-free mice, and potential product porphobilinogen. Then we used genome scale enzymatic network analysis to identify enzymes connecting potential substrates to potential products. For example, here, 5-aminolivulinate can be connected to porphobilinogen by a single bacterial enzyme, phosphobilinogen synthase, as part of the heme biosynthesis pathway. In this manner, we scanned all possible substrates and products from this scatter plot and found 93 pairs that can be connected by a single enzymatic reaction, and over 400 metabolites that were connected to an annotated metabolic enzyme of bacteria. So before we can identify microbial activity explaining these metabolic differences, we first have to investigate what actually happens to the microbiome under these two different dietary conditions. And the first observation that we made is that their microbial composition in the mouse is quite stable between the mice and between conditions. And as shown in these bar plots, other fractions of different microbial species from our community that we assembled in control mice, mice fed high carbohydrate diet here in blue, and high fat diet here in red. And as you can appreciate, all the Bacteroides species in red are the major phylum in the mouse gut in our experiment. When we plot their normalized microbial abundance across the conditions and mice in this heat map on the right, we see that there are consistent changes between microbial abundance, such as microbes belonging to the phylum Bacteroides are more abundant on the high carbohydrate diet, and microbes that are members of the phylum Firmicutes, Actinobacteria, and Proteobacteria are more abundant on high-fat diet. We also analyzed transcriptomic changes in microbes in either high-fat or high-carbohydrate diet. And what is shown in this bar plot is the fraction of genes that are changing between the two diets. And as you can see, all the microbes are regulating their gene expression, and some microbes have up to 10% of their genes either up or down regulated, either in a high carbohydrate or high fat diet. So to describe this transcriptional changes of the microbes, we performed pathway enrichment analysis shown in these heat maps on the right. And as you can see that, for example, metabolism of microbes on high fat diet here in orange is characterized by more activity in amino acid biosynthesis pathways highlighted here in yellow, and metabolism of microbes on high carbohydrate diet is characterized by more activity in starch and sucrose metabolism, and also more activity in fatty acid biosynthesis, suggesting that on high carbohydrate diet, microbes have to perform fatty acid biosynthesis, whereas on a high fat diet, potentially that they can reduce this biosynthesis because that can take up fatty acids from the diet or from the host. So to connect this microbial adaptations in the species abundance and microbial transcriptional activity in the genes to our results from the metabolomic screen, we performed the analysis of variants, and for each metabolite calculated how much variation in metabolite abundance can be explained either by species abundance or by species gene expression. And in the scatter plot on the left, I'm showing you each dot is a metabolite, and on the x-axis, I plot the R-squared value or the amount of variance that can be explained by species abundance. And on the y-axis is the amount of variance in the metabolite abundance that can be explained by gene expression. And we found that together about 30% of metabolites in the gastrointestinal tract can be, the variance, or 70% of their variance can be explained either by the changes in species abundance or changes in gene expression in bacteria, or both. And to give you some examples, here the porphobilinogen, a metabolite that I showed you before that was a potential microbial product, is the variance in porphobilinogen abundance can be explained by the variance in abundance of 5 bacterial species, 2 of the genus Actinobacteria here in green, and 3 of the genus Firmicutes here in blue. This plot shows how their relative abundance of bacteria is associated with the relative abundance of this metabolite in vivo in the mouse gut. But more importantly, when we look at the gene expression, at the expression of porphobilinogen synthase that we identified with the network analysis, we found that in Clostridium scindens, the expression of this gene was highly correlating with the amount of produced porphobilinogen, suggesting that this bacteria probably is the main player responsible for the differences in porphobilinogen abundance in the gut of colonized mice. So with this, I want to summarize that our computational and experimental approaches allowed us to perform unsupervised clustering of metabolite profiles across the mouse tissues that revealed that there are stable in vivo tissue metabolite profiles independent of the mouse colonization state and the mouse diet. On the other hand, our simple metabolic flux model allowed us to quantify diet, host, and microbiome contributions to the metabolites in the gastrointestinal tract, and suggest that about 40% of the metabolites are contributed by the diet, 30% are produced by the host, and at least 15% are directly dependent on the microbiome presence. Finally, integrating metagenomics and metatranscriptomics data with metabolomics help us to identify potential bacterial players and their genes that can explain and potentially predict the differences in metabolite abundance in the mouse gut. With this, I would like to thank Andy Goodman's lab at Yale University, where I started this project and performed all the experimental work, and my close collaborators, Tasha, Rebecca, Whit, and Michael, and the Borg lab at EMBL Heidelberg, where I'm based now and where I'm finalizing now the diet project with the help of the close collaborators, Renato, Michael, Suguru, and Sander. And I'm also thanking, of course, my funding sources, and I thank you for your attention, and I will be happy to take any questions.
Thank you so much. That was very enlightening and very full material. So that was really fascinating. I think from here, Arpana can take on the questions and steer the discussion. Arpana?
Yes, of course. I'm happy to. Thank you, Dr. Zimmerman, for that really wonderful talk. Like Oliver said, you really had, a question for each topic, and really loved your approach on how you asked those questions and answered them. So there's a few questions in the chat that I'd like to go over. When you were talking about what determines bacterial survival in your earlier part of the talk, there was a question, since Ppgpp is involved in replication, is it possible that during carbon starvation experiments, does the Ppgpp deletion mutant become non-culturable? And the regulation of sugar and amino acids is a hallmark for a non-culturable switch to non-culturable in bacteria. Did you investigate those options?
Yeah, thank you. It's a great question, and I mean Ppgpp is really like a global regulator. So indeed, a lot of processes are involved. And what we believe is the major course of Ppgpp0 phenotype in vitro is that the lack of Ppgpp molecule actually makes bacteria lacking a signal to stop growing. So we believe that, so basically Ppgpp, when it's produced, it induces a growth arrest in the cells, in the wild type cells, and the Ppgpp0 strain is not able to arrest growth and continues to waste resources. And of course, I mean, alpha-ketoglutarate is only one part of the potential mechanism of how this is regulated. And there are many more mechanisms to discover. And I do agree that it can be definitely looked more into, like amino acids and sugar starvation, sugar regulation more in depth.
Great, thank you. During your second part of the talk, specifically on answering how bacteria contribute to host metabolism, there was a very interesting question that I got regarding to screening drugs. Is it possible to screen different drugs according to their efficiency in patients based on their microbiota?
This is a fantastic question. Nowadays, more and more studies report cases where also in humans, we can state that microbiome actually determines the efficiency or efficacy of drugs. And I think there is still a long way between our very targeted and controlled in vitro studies and human applications. But there is definitely a lot of interest and a lot of work into that direction, that basically in cases where we know that microbiome can affect drug metabolism, and we know what microbes or what genes determine these differences, we can actually screen patient stool samples before defining a therapy and choosing a drug dose or choosing a combination of drugs in a more informed way based on what microbes are present.
Wonderful. I think during this section, you had a slide that showed gain-of-function libraries. There was a question on how they're created, and I'd actually kind of want to know what is a gain-of-function library?
Great. So I very briefly went over it. So gain-of-function library is a genetic, is an approach in microbial genetics where we try to identify parts of genes or parts of genomes that have a certain function. And in this case, we have our bacteria of interest, for example, Bacteroides tetraiota omicron that I was showing in our study first. And we know that this bacteria can metabolize a drug, but we don't know what gene is responsible for it, right? So to try to identify what gene is responsible for this drug metabolism, we can shear the whole genome of the donor bacteria into small pieces and test each of these pieces whether it actually can metabolize the drug. And to test this, we express these parts of the donor DNA in E. coli, which is a different bacterium. It's a very common laboratory bacterium that is very popular for genetic studies. And E. coli alone is not able to metabolize this drug. But then we insert the little piece of DNA of the donor bacterium in this E. coli strain, and then we test whether it gained the ability to metabolize the drug. That's why it's called gain of function, right? So when we express a random part of the donor DNA in the E. coli, we test then whether E. coli gained a function to metabolize this drug. And then then we have like several thousands of E. coli strains. We don't know what part of DNA is expressed there, but we can functionally screen and find those that did gain the function to metabolize drug. And then we can sequence what is the expressed donor part of donor DNA, and then map it back to the genome. And that's how we identified which genes from the other bacteria, when expressed in E. coli, made it metabolize the drugs of interest.
That's really interesting, and definitely a key component in integrating, multi-omics data like you've presented here today. We have a lot of interest in terms of the bacteria and in the context of diet. I mean, that's definitely something very interesting to learn about. There's a question here. A recent study from Sanger and Embol also identified 140,000 virus genomes in the human gut. Can you tell us more about viruses in the human gut and how this can influence future microbiome studies?
That's a great point indeed. In my research, we focus on bacteria, but there are also fungi and viruses and other microorganisms in the human gut that can play a huge role. We do not know, or I don't know, whether viruses can play a role in the drug metabolism, for example, but they definitely shape, they can play a role in how they shape the microbial composition. And I think there is a lot of viral research that is yet to come and to help us understand and also control microbiome composition in the host.
Yeah, I definitely agree with you. I mean, nowadays, we're definitely going to see a lot more research on the impact of virus and especially the impact on the bacteria itself. I had a question for you in terms of the types of diets, because you had covered high fat diet and high carbohydrates. Are there similar contributions to host metabolism in other types of diets? For example, paleo, vegan. I'm a vegetarian myself. Like, what can I expect? So,
I mean, that's definitely true that microbiome plays a role in any diet. And there are a lot of studies also in humans that show that different types of diets are affecting the composition of microbes, right? Because many microbes in our gut are specialized in metabolizing certain type of nutrients. There are metabolites that are more specialized in metabolizing starch and complex polysaccharides. There are microbes that are more specialized in metabolizing meat, or some that are strive in fat diet. And there have been also a study already about 10 years ago that had a very nice by Jay Faith, and they had a very nice system where they had like one macronutrient in the diet, and they had, I think, 8 diets or so, and they could predict based on which nutrient is the major calorie contributor in the diet, which microbes will strive. So there are definitely a lot of relationships between diet and microbes, that both affect the composition of microbes, but also what they're doing, right? How they adapt and what are the pathways that they're activating or deactivating. In our study, we were focusing more on like, can we identify, can we quantify for each metabolite where it's coming from, and can we put a name on the bacteria that is potentially contributing to it? But there is more to come, and I'm sure that like if we looked at more diets, then we would see more variety of responses.
Thank you. There was actually another question specific to the high fat diets, and there was a question there. It says, it seems that the metabolic shift went from sugar to amino acid metabolism with the high fat. Did you find a change in the ppgpp in this diet shift? That's always a mouthful to say.
That's actually a really interesting point. I have not I have not looked at the ppgpp related changes in the microbes. I will definitely do it and check whether there is any evidence for ppgpp response. I think, I mean, I wouldn't, so our hypothesis from the ppgpp project is that ppgpp is needed, like that microbes evoke this response at different times during their colonization, but it's not like always on or always off. So we expect some fluctuations, like when the food is abundant and the host just ate, then the microbes are thriving, but then there is a period of no food, and then the microbes need ppgpp response to kind of arrest their growth and wait for their food to come. So I think that the shifts between the sugar and carbohydrate metabolism to amino acid metabolism is rather showing different types of foods that they encounter at different diets, rather than, you know, stress imposed by the lack of food. But it's a hypothesis, and I think it's a valid hypothesis, that some microbes that some microbes can be stressed by the lack of amino acids, for example. That's also one of the hallmarks of ppgpp response.
Yeah, and since everything is such a close, network of things, one plays a role in the other, it's definitely a valid hypothesis there. We have a question that's a little bit more on the analytical side. You had reported that there was 35,000 metabolites within your study. How many LC methods did you combine to get such huge metabolite coverage? And did you annotate these metabolites based on MS1 only, or are you also using an MSMS library? And is that library in-house?
So to answer the second part, it's MS1 only. So it's only time of flight mass spectrometry. Basically the annotated library is not in-house. It's a publicly available combination of KEGG metabolites and human metabolome database. So I combined the huge list of metabolites and annotated with that. And it's MS1, right? So it's based on mass over charge. To answer the first part of the question on the analytical method, we did use 3 different columns and 2 ionization modes. So this data was generated with both in positive and negative mode, with a high-leak column, C8 and C18 columns. So you kind of can say 6 different methods, and then pooled together, filtered for like quality control, and then used to basically map against the annotation database.
Thank you on that one. I had a question, I guess it's more about your process. What methods do you use to integrate your multi-omics data? You were showing, I think, at least 3x3 in some of your slides with RNA and metabolomics and all that stuff. Do you have a preference in software or strategy? Did you develop something in-house, or are you using commercial? How are you integrating that data set?
So the integration methods are, in my case, usually project specific. My kind of umbrella method is to integrate via metabolic networks. And for that, I'm using, for example, graph-based approaches where with some in-house code that I believe is available, and if not available, it will be available when the diet project will be out, where I'm basically just using standard approaches to identify metabolic paths between the different metabolites and integrate in this way metabolomics with transcriptomics or metagenomics. There are also a number of methods that I've been trying out that are more dimensionality reduction methods that are taking all their omics data into as input and then producing a factor analysis identifying what factors determine the differences between different conditions, but it depends on the research question. So yeah, I mean, if someone has a specific question, just drop me a line. I'd be happy to chat and to share my experience. But I don't have a preferred software for integration.
We definitely have one grad student who's answering those questions now, so I'm sure she'll drop you an email. There was one last question in the chat, and I'll hand it off to Oliver. Maybe he has a few questions. The question was regarding sharing the references for the modeling of the diet with metabolites and microbiome. If you would share your references there.
Okay, so
yeah, the question was, do you mind sharing the references regarding the modeling of diet with metabolites and microbiome?
I of course don't mind. So just, I guess after the talk, right? Or do I get the question correctly?
Yeah, I guess
just like really drop me a line, and I'm happy to share anything I can share. So
okay.
That's
And then there's one more question that just came up in terms of software. Is there a specific software for factorization analysis for multi-omics data?
Well, I can advertise the one from EMBL, which is MOFA2, for example, multi-omics factorial analysis. But it really depends on your research question, because the factorization analysis usually focuses on identifying the most informative features in your data set, whereas sometimes the question is to try to analyze as many as possible. Like in our metabolomics approaches, we try to go broad, right? And we try to see, okay, other chemical groups or other KEC pathways or HMDB pathways that are associated with either one or the other condition or metabolic profile.
Great. I am sure you can find that link on the EMBL page. Oliver, would you like to chime in?
I mean, usually we would now have the day and discuss more things. I have some simple questions, like you showed k-nearest neighbor clustering approaches. When you do that, you have to say how many classes you define, and how do you approach that? I mean, do you take 5 or 10, or what is your number of classes in this instance?
Yeah, I mean, that's a very good point for k-means clustering. I mean, it's unsupervised, but you have to provide k, and of course, k will determine the outcome. So for this, to try to make it as unsupervised as possible, I first ran a analysis that identifies the best k based on how. So to do a grid of k search between 1 and 10 or 15, and for each k calculate the differences, the within cluster differences and between cluster differences to determine the k at which those differences are the biggest. I mean, there are several methods to determine the optimal k, for example. And in this case, I ran this analysis several times, and the optimal k was between 5 and 7. So I choose 6. But then what I also do, since their k-means clustering also depends on your initial choice of cluster centers, and it can slightly differ. I also run this analysis then several times, usually several hundred times. And then for each metabolite, I record in which cluster it was associated to. And then I did have cases where metabolite was always in the cluster 1, so it was a clear case. But there are also metabolites that are half of the time in cluster 1, half of the time in the cluster 2. But at least this unbiased selection of K based on a certain criteria and running it multiple times to make sure that you have a robust, or at least you can give an estimate whether it's robust or not, depending on how many times each metabolite was attributed to each cluster.
So when you do that, you have range scaled your metabolite intensities. And in order to do range scaling, that's always a little tricky because, you know, a drop of 10%, you know, may appear after range scaling just as big as a drop of 10-fold. And then it becomes the question, how do you distinguish something that is circumstantial or something that had actually a big effect size in these manners? So a little bit like, how did you curate your data before, weeding out compounds that had low precision, you know, like, you know, let's say 30% coefficient of variance in multiple pools, or what do you do there to make sure that you only get metabolites into your data set that have at least a very good analytical reproducibility?
Yeah, I mean, we do a number of filtering, of metabolite filtering at different steps of analysis. So we start with basically just the noise correction and just removing those that are not found in that many samples, right? Like that fraction of samples. I mean, that's true, and that scaling is always dangerous. I mean, we also do then like quantile normalization of all samples to make sure that at least we correct for potential batch effects between the different samples, so that we have the same distribution of intensities. And then, so I think that for spatial metabolite abundance analysis, that it's not that critical that we are scaling metabolites of very different intensities, because we are interested in the spatial profile across the tissues, and then it's more like the shape than their magnitude of difference. But then for metabolites that, so then we also do like more regular differential analysis between metabolites and the tissues, and then we take into account their variability and their intensity values. So we don't normalize when we compare metabolite abundance in one tissue versus other in the two conditions. And I mean, we do have 5 replicates, which is, it's a question whether it's good, but it's better than usual 3. And we have we usually have like a quality check that the variance in the replicates is within a certain range.
Okay, that's good to hear. Now you mapped ions to potential metabolites and databases. Did you also use ions that did not map anywhere, so unknowns? And what did you do with those unknowns then?
So for the diet project, it's still ongoing. So what I was showing you today, it's only the ones that we could map. But I'm also now performing analysis on the unknowns. And I mean, we take them. So I mean, it's a question like what do you want to achieve, right? So we can take them into account in the analysis and analyze as everything else, and just then do like interpretation only of those that we can annotate. And I see, at least for the analysis that I showed you now, like for their spatial distribution and their kind of metabolic modeling of their different sources, I see that it's pretty much very similar results, so very similar fractions. Also, if we take all the unknowns into account, with the drug metabolism projects, unknowns was our pearl, right, in a way, because we knew the drugs that, I mean, basically we did a very thorough filtering of unknowns to identify those metabolites that we were sure that, or at least with a certain, with a certain level of sureness, we could say that those are drug metabolites, and there the unknowns was our gold mine, basically, to identify the chemistry.
Yeah, the deacetylases. All right. Now, when I look at all the work, it's clear that it's a teamwork, right? I mean,
absolutely.
You know, people who've done the libraries of the gain of function, and people who do the screens, and people who do the mass spec, and people who did, you know, so many things. And we see some of those heads here in these photos. But if I understand correctly, you did most of the data analysis, right? The
yes.
network analysis, but you also did data curation, data cleaning, or did you just get a clean data set from somebody who says this is what it is?
I mean, I do most of the curation myself, and I mean, as you said, it's a teamwork, and it's also like those 4 people from the Goodman lab are the main contributors to this team effort. So we also had a very good luxury position where we could talk. You know, it's not like we send our samples somewhere and then we get the table back. We generate them ourselves, and then we discuss, like, okay, what are the curation steps that make sense or don't make sense. And for example, with the drug, with the analysis of untargeted drug metabolites, we did like a multi-step filtering procedure, because we had several thousands of metabolites that were potential drug metabolites, and then we did like a 5-step filtering steps based on the mass shift of the drug and also the correlation with the parent compound. We also found that some drug metabolites were actually mass spec artifacts. So they were basically a product of ionization, right? You shoot the drug into the machine, and then deacetylation happens, for example, just chemically without the presence of bacteria. And those artifacts are actually quite common. So we also checked for example, if the drug metabolite and drug have the same retention time, likely their chemical conversion happened already in the machine and not in their test tube. So there is a lot of steps involved in data curation, and we try to describe them as good as possible when we publish, but we're always happy to discuss, of course.
All right, thank you so much, Dr. Zimmerman. Do you have any questions for us?
Are you? Well, so maybe one thing that I don't know if we are struggling, of course, always with measuring certain classes of metabolites, for example, lipids and fatty acids that are very hard to cover with our mass spectrometry pipelines. So do you use different approaches to measure those?
Yes, like, we use fractionation. So we use the Mathias or modified Mathias method so that we have the lipid fraction as one part. And then we use Waters columns, either the BEH or the CSH, for separating lipids. We've had good success with those. You know, but of course, also in lipidomics, there are lots of unknowns. And we wonder, just as you did, how many of those ions, where we don't know what they are true, and how many are either noise or are in-source fragmentations. And that is ongoing work. But clearly we think that going beyond discovery work needs to go beyond what is known. So if we only rely on KEGG maps and on HMDB, that's very dissatisfying, right? And it can be wrong because, you know, mapping an ion is, well, sometimes you have multiple isomers, of course, and sometimes it might just be wrong. It might be an adduct of something else, or even a completely different thing. So that nobody can answer that, of course, how many of those are correct. And we have been incorrect in the past. So we put our data out. So like for the Metabolics workbench or other databases, so other people can scrutinize that. And do you also do that? Like, you know, putting the data to public websites like, I don't know, Metabolites or anywhere?
Yeah. So for the drug screen, we did put all the data to Metabolites, which was a very painful process, but I think also because of the amount of data we generated with all this screening of libraries and basically just even if you take their 70 times to 100 always over time already makes it difficult for Metabolites, but we worked with them like to make sure that we can upload our data there. But yeah, I think that's definitely something in Metabolomics community that is not as well structured and defined as in, for example, sequencing, which is much easier to standardize.
Well, one of the things is that we don't have standardized retention times. And standardized
exactly.
Processes. So, you know, we have just embarked onto an unknown lipidomics consortium work group where we used the same samples in different labs and used both common methods and in-house methods, and just to see how much overlap we get and how much harmonization we can get when we put our data out. And then we did that first for lipidomics. Maybe we'll also do it for polar compounds too. So that is definitely something that would need to be improved. But on the other hand, at least in the United States, at least those people who are still on the Zoom link, which is about half of them, good for us. We go fund by fund, so we are not like an EMBL, who are funded by a national or supranational organization. But we go from one project to the other, and each project is usually too small to build these resources, right? Yeah. Whereas EMBL has the benefit of having, you know, the big organization over in Hingston and Cambridge, UK, as well as, for example, EMBL in Heidelberg, right? That is has a trajectory where you can do these kinds of database developments. So we hope that Metabolites will evolve and you know, lead the way perhaps at some point. But I agree, getting data sets into Metabolites is difficult. It is maybe a little easier to get them into Metabolomics workbench, but it's not easy to get them out, in the sense of, you know, saying, okay, give me everything you have on microbes, or everything that is related to gut microbiome, or gut microbiome drug interaction. These questions are usually not organized in that way, so that you can have an organizational taxonomy, almost ontology on top of the data set, so that you can easily go through the different data sets and then reuse them, or even try to align them. That was not done either in Metabolites or Workbench. But hey, what can I say? We are certainly not going to do it.
Yeah, let's see. I mean, we're now trying to push Metabolites. Well, it's a long process, but at least to talk to the developers and managers to get it more standardized and more easy to use and more like high throughput and large scale.
Yeah. From the 56 participants, are there further questions that people are burning to ask and didn't dare to ask so far? people are still hanging on and hope to get these golden words of wisdom from Dr. Zimmerman. So that's not the case. Well, then thank you so much for your time. We really enjoyed your talk. Good luck with your research, and hope we will see each other at a conference or another venue at a given time. Thank you so much.
I hope so too. Thanks a lot for inviting. It was a big pleasure.
Okay, bye-bye.
Have a good day.